Thank you, PEROSH, for giving me this opportunity to present a part of my PhD thesis, which is the network in co-environment. And I hope through my presentation, I can provide the scientific information why it's not okay to perform the network in co-environment in the molecular way. Because like we are living in the world due to the development of information, communication, and technologies, it's not difficult to travel across the world. So it's natural that the employers want to increase the staff around the clock to produce and distribute the products in different time zones. According to the figures from our institute, 8% of the Norwegian workers self-report a daily exposure to the cold during their most of working time, and 4% work regularly at night, and 10% work occasionally in the Norwegian working population. And among the workers that are exposed to the cold during the time working hour, they also report the non-daily time working hours as well. And here are two examples that the workers have to expose to different occupational factors, for example, the fishery and then the construction workers. So that's why it is logical to assume that occupational cold exposure at night is quite common now. And according to several studies, it indicates that cold exposure can lead to reduce the core body temperatures, which is called hypothermia, or it can leave the skin but permanent skin damage, which is called turbulence. And in addition to that, night shift also have the detrimental effect on the cognitive functions, thermoregulation, cardiovascular diseases, metabolic health, and muscle skeleton diseases. And the night shift have this bad influence on our body health because it disrupts this circadian rhythm pathway. And this circadian rhythm pathway it starts with the light in the upper left corner, and the light we receive from the sun naturally, and this light information will be transferred into the photoreceptor in the eyes, in the retina of the eyes, and this light information will be transmitted into one brain region called the suprachiasmatic nucleus, which is a part of the hypothalamus. Hypothalamus is the brain region that is responsible for regulating the homeostasis for our body, for example, regulating the body temperature and something like that. And from there, it will transfer the information to different brain regions, for example, the hippocampus for the cognitive function, or pineal glands to regulate our melatonin. So that's why the melatonin usually very high in the evening because there is no light at all, but it's gonna be stopped at around 7 a.m. in the morning because that is when our body receives the light. And from the central nervous system, this light information will be transferred throughout the body through the backbone and some internal organs as well, for example, edible tissue, the lungs, the heart, and the intestine. So that's why nearly all the tissues have their own biological clocks, and this results in different organisms. And in order to maintain this pathway, our body needs to have some kind of genes which is on the right side of the slide, called the BMO1 and the clock. This protein will be highly expressed in the morning because of the light, and this will induce the impression or activation of different kind of genes. For example, the PER and the GRI, which is very highly expressed in the evening. But after being turned into the protein, PER and GRI are going to bind together, and they inhibit the BMO1 and the clock by themselves. And this cycle takes 24 hours to finish, and it's not supposed to be broken or modified. So that's why that's been to our presentation, the result question. Does the stimulus exposure to the cold working environment and night shift hold any relevance for workers' health? And does the thermoregulatory response differ according to the time of the day? And in order to answer to these two questions, we're going to use mouse model to perform the experiment. not human. The reason why was because mouse and human are under their different species, but they share very similar genome. So that's why when our body receive or the mice receive the cold, it will trigger signaling pathway, and it will eventually release the norepinephrine, and this will bind into their own receptor on the edible tissue, and eventually it will increase the UCB1. UCB1 is the thermoregulated biomarker, and with the cold exposure, it will not be released. So that's why we're going to study about this protein. In order to do that, as I say, we're going to use mice, and the mice have been acclimatized for 27 at the 27 degrees for 1 month, and to let them get used to the living environment. And after 1 month, we performed the cold exposure experiment on them at different times of the day in 8 hours. And after performing the cold exposure experiment, we're going to sacrifice the mice and take out the brown edible tissue. As you can see, this is the different stage of the brown edible tissue before and after cold exposure. It's become more brownish because of the activity of UCB1. And so that's why we measure the UCB1 mRNA and the protein levels. And there is a clearly significant difference no matter the time of the day at the mRNA level. But when it comes to the protein 1, surprisingly, as in the morning, there is no difference at all. But in the morning and during the night shift, there are a significant difference between the protein level. We also check whether the co-exposure also affects the expression of the core clock genes, the B1 and the clock, and they also affect to all of the clock genes. So that's why we want to conclude that the timing of co-exposure matter, because the level of UCB1 mRNA and proteins show distinct variation depending on the time of the day, and the expression on the core clock genes was assumed to be modified in the mice subjected to co-exposure compared to the control one. Yep, thank you.
Thank you very much.